So like here in this slide, you can see here that suppose we observe a pleasant pursued by a fox. So this scene has obvious evolutionary and ecological importance. If the pleasant is to continue passing its genes to future generations, so it must win the contest actually. But if it loses, the fox gets to eat. In other words, the outcome is significant. But what determines the outcome is actually the physiological properties of many of the pheasant's internal tissues that play the major roles in this. So speed at which the pheasant can escape, for example, depends on the precise ways that the pheasant's muscles work. So as they contract to produce the mechanical forces that propel the pheasant forward, the pheasant's speed also depends on its, here you can see, it depends on its chemical properties. That means they contract to produce the mechanical forces that propel the pheasant forward. So pheasant speed also depends on the biochemical mechanisms of producing this adenosine triphosphate. Because the muscles that use this adenosine triphosphate as their source of energy for producing these mechanical forces, and consequently they can work only as fast as their ATP supply permits. So this ATP, adenosine triphosphate, is actually the component of energy that gives them the energy to escape or to do the mechanical, to do these mechanicals, to do these this biochemical mechanism. So in a very real sense, the pheasant's success at the ecological and evolutionary level of organization, it's actually a direct consequence of physiological capabilities at the cellular and biochemical levels of organization in its tissues. So here we can say that it actually depends upon this ATP production that plays a key role to escape from a predator. So talking about the physiology of fish, that includes the evolutionary biology, the laws of chemistry and physics, the drawing in system physiology, which is a cross-section of the body, the ecological relations, as well as the organization. So mechanism actually refers to the components of actual living animals and the interactions among those concepts that enable the animals to perform as they do. So since we all know that a physiologist typically begins an investigation by observing a particular capability that excites the curiosity or needs to be understood for practical purposes. For example, the capability of human visual system to distinguish between the red and blue can be an example. So another example can be the ability of certain types of nerve cells to conduct nerve impulses at the speeds of our 100 meters per second. So whatever the capability of interest, the typical goal of physiological research is to discover its mechanical basis. So what cells, enzymes, and other parts of the body are employed and how they are employed to enable the animals to perform as it does. So that's the need of that physiologist. So for a detailed example of mechanism, let's consider the example of fireflies. For example, how the light is produced by a light organ at the posterior end of the abdomen. So each light producing cell in a firefly's light organ receives the molecular oxygen by way of a branching system of gas transport tubules. So this oxygen actually enters the system from the atmosphere and is carried to the tubules, to all the tissues of the insect's body. So to understand how a light producing cell emits the flashes of light, let's start with the biochemistry of the light production. So actually what happens is that a chemical compound, a benzothiazole named the firefly luciferin, first reacts with the ATP. What it forms is the luciferyl adenosine monophosphate. Then if the oxygen can reach this luciferyl adenosine monophosphate, the two react to form a chemical product in which these electrons are actually boosted to an excited state. And when this electron gets excited, the product returns to its ground state. So it emits some photons. So this sequence of reactions requires a protein catalyst that's called the firefly luciferase. So a question only recently answered is how these cells within the light organ are controlled. So that they flash at certain times but remain dark at others. So when a firefly is not producing light, like here you can see. So this is actually, this is called the lantern, the light organ, the lantern. What actually happens is that since we already discussed that they have some kind of an enzyme called the firefly luciferase, so that helps them in lighting up. So, and when the oxygen enters their body, so they just flash, and at certain times they remain dark at others. So when a firefly is not producing light, any oxygen that reaches the insect's light cells via its gas transport tubules is actually intercepted, and thereby preventing them from reacting with this luciferyl adenosine monophosphate that is secreted by the mitochondrion. So they are positioned between the gas transport tubules and the sites of the luciferin reactions. So light cells produce light when because of stimulation by the nervous system. So mitochondrion become bathed with nitric oxide, and nitric oxide blocks this mitochondrion use of oxygen, allowing this oxygen to react with luciferyl adenosine monophosphate. So these facts actually, facts like these form a description of the mechanism by which the fireflies produce this light. So another topic that we will be talking about in this chapter will be the conformity and the regulation. So I'm trying to record this PowerPoint, record this lecture so that other students can also have this. So internal environment may be permitted to change when the external environment changes, or it may be kept constant. For example, the animals have evolved various types of relations between their internal environment and the external environment. If we think of the organization of the body as being hierarchically arranged, the relations between internal and external environment represent one of the potential hierarchical levels at which animals may exhibit organization. So actually the animals, they display the two principal types of relations between their internal and external environments. On the one hand, when the conditions of outside an animal body changes, the animal may permit its internal environment to match the external conditions and thus change along with the outside changes. On the other hand, the animal may maintain the constancy in its internal environment. So these alternatives are all illustrated in this figure you can see here. So this is all about the A figure is about the temperature conformity and the B is about the temperature regulation. So what happens is actually if the temperature of an animal body, animal's external environment changes, one option, that's for the animal to let its internal temperature change to match the external temperature. The another option is for the animal to maintain a constant internal temperature. You can see in the figure B. If an animal permits this internal and external conditions to be equal, it's said to be show conformity. And if the animal maintains regulation, the conformity and regulation are the extremes. So there might be some intermediate responses that will be common. So another topic that here, the heat acclimation in humans as measured by the exercise endurance. So how it actually works. So this is actually the mixed conformity and regulation in a single species. So here you can see that individual animals that are subjected to a change in their external environment, they exhibit acute and chronic responses to the environmental change. These acute responses are actually the responses that are exhibited during the first minutes or hours after an environmental change. Or there might be some chronic responses that are expressed as following prolonged exposure to new environmental conditions. So the acute responses, they actually exhibit the acute and chronic response to the environmental change in no time. And in chronic responses, it might take even minutes or hours after an environmental change occurs. So chronic responses are expressed following prolonged exposure, we can say that. So talking about the heat acclimation in humans, which is measured by the exercise endurance. So for example, here in this graph, you can see there is a 24-feet young man without recent heat experience were asked to walk at 3.5 miles per hour in hot. So dry air was like 49 degrees centigrade. Their endurance was used as a measure of their physiological capability to engage in moderate work under hot conditions. So this acclimation is illustrated by the chronic response is reversible. So if heat acclimated men return to a life of no heat exposure, they gradually will revert to the level of endurance that's evident on the day one. Here you can see in the graph. So it was seen that this is the way of quantifying their physiological ability to sustain this moderate exercise under the hot conditions. So none of the men actually had sufficient endurance to walk for 100 minutes on the first day. However, as the days passed and the men had more and more experience with these hot conditions, so actually they get adopted. So their endurance increased as indicated by a steady increase in the number of men who could keep walking for 100 minutes. So this endurance measured as number of men who could walk for 100 minutes, it's increasing here. So as soon as they get acclimated, or we can say adapted to these environmental conditions, so their days of exposure, it also increased, and then as such, the number of men also increased. So this is all related to the physiological functions of the animal body. So here in this table, you have the 5 time frames in which physiology changes. For example, we talked about the acute changes. Those are the short-term changes. And chronic changes are actually the long-term. It can be minutes or hours when they are exposed to the external environment. So evolutionary changes we have that occur by alteration of gene frequencies over the course of multiple generations in populations that are exposed to new environments. So these changes in physiology that are internally programmed to occur, whether or not the external environmental change, there are some developmental changes and some changes that are controlled by the periodic biological clocks. So when talking about the developmental changes, these are the changes in the physiology of individual animals that occur in a programmed way as the animal matured from the conception to the adulthood. The changes that are controlled by the periodic biological clocks, these include the changes in physiology of the individual animals that occur in repeating patterns, for example, each day under control of animals. So these are the internal biological clocks that provide these changes. So moving to the butterfly biogeography, how the species diversity is relatively low at the cold, or we can say how this species diversity is relatively low at the cold or high altitudes. So environment has a main role in predicting these physiological functions. So what actually an environment is? An important starting point in answering this question is to recognize that animal and its environment are actually interrelated, not independent entities. So they are in fact defined in each terms of each other. So environment in particular use cannot be specified until the animal is specified. For example, we can take an example of dog is an animal from our usual perspective. But if the animal of interest is a tapeworm in the dog's gut, then the dog is the environment for that tapeworm, right? So all animals, in fact, are parts of the environments of other animals. The birds in the trees around your home, they are part of your environment and you are part of theirs. So this interdependence of animal and environment is reflected in standard dictionary definitions. So what actually environment means? So an environment is defined to be all the chemical, physical, and biotic components of an organism's surroundings. So if we talk about the temperature, that's a part of environment. So temperature of the air, water, or any other material, it's a measure of intensity of random motions that the atoms and molecules in the materials undergo. So all atoms and molecules, they ceaselessly move at random on an atomic molecular scale. For example, a high temperature, it signifies that the intensity of this atomic molecular agitation is very high. So most animals are temperature conformers, and we discuss temperature here. We will focus on them also. The conformers are our principal interest because the level of atomic molecular agitation in their tissues, it matches the level, the environments where they live. For example, here we will discuss about the butterfly biogeography species. So the diagram here shows the number of species of the swallowtail butterflies at various latitudes. So the reason there are relatively few species of animals at high latitudes may not be simply because of the low temperatures there, but maybe in part because of relay effect from the effects of cold on the plants. So plants decrease in diversity and animal productivity towards the poles, affecting the food supplies of these animals that decreases the number of these butterflies. So that means here what we are discussing is that this temperature has actually, so these are interdependable. So the temperature here in the polar regions that have an effect on the diversity of these butterflies, or we can see the biogeography of these butterflies. For example, for greater extremes of cold that are found on land in aquatic environments in Antarctica, the air temperature sometimes drops to minus 90 degrees centigrade in the Arctic, and it descends to minus 70. So the extremes of this animal adaptation to low this tissue temperature are represented by certain extraordinary species of Arctic insects that spend winters inside exposed plant stems on the surface of these pack ice. So these insects are in a state of suspended animation at these times. So the diversity of terrestrial temperature conforming animals typically becomes lower and lower as the latitude increases from the temperate zone towards the poles, as we have seen in this figure. So this decline in diversity towards the pole indicates that very cold terrestrial environment are demanding places for animals to occupy, despite the evolutionary adaptability. So in short, we can say that these butterflies and the temperature in these poles, they are in short, the environment. So these are actually interdependable. So they depend on each other. For example, we have another example, the hottest places in the biosphere are the waters of geothermally heated hot springs and underwater hot vents. So these waters are often far above the boiling point when they exit the Earth's crust. So although aquatic animals, they typically stay where the waters have these, the boiling, these uh, temperatures. So we have here one more example of the thermophilic lizard. Like the name implies here, thermophilic means the heat tolerant, or we can say the lizard that this thermophilic lizard, it's most common in North American deserts, the desert iguana. And the zoological name of this is Dipsosaurus dorsalis, that can often be seen abroad as the sun beats down on hot days. So although it doesn't usually expose itself to body temperature higher than 42 degrees, it can survive until 48.5 degrees centigrade. So one of the highest body temperatures tolerated by any vertebrate animal. So this is actually the thermophilic, that means heat loving lizard. Thermophilic means the heat loving. So one more here is the performance in an oxygen poor environment, how the oxygen affects is depend, how the animals depend on the oxygen. So need of most animals for oxygen is a consequence of their need for metabolic energy. We all know that chemical reactions that animals use to release energy from organic compounds, they remove some of the hydrogen atoms from the compounds. So each adult person, for example, liberates 1/5 of a pound of hydrogen every day in the process of breaking down food molecules to obtain energy. So this hydrogen actually, which is liberated, cannot be allowed to accumulate in an animal cell. Thus, an animal must possess biochemical mechanism for combining this hydrogen with something, and oxygen is the usual recipient. So this oxygen obtained from the environment is delivered to each cell. What happens? It reacts with the free hydrogen products in the cell, yielding water. So suitability of an environment for animals often depends on the availability of oxygen. For example, here consider an example of high altitudes. They are among the Earth's most challenging places, reflected in the fact that number of animal species are sharply reduced. So at such altitudes, the maximum rate at which the animals can acquire oxygen is often much lower than at sea level. So, and functions are also limited. So at elevations above 6,500 meters, or we can say 21,000 feet, for example, people breathing from the atmosphere find that simply walking uphill is a major challenge because their level of exertion is limited by the low availability of oxygen. We all know that as we go higher, so it becomes very, so you're walking uphill, it's a major challenge actually because their level of exertion is limited by low availability of the oxygen. So some animal species have evolved adaptations to succeed in the dilute of oxygen of these air in ways that humans cannot. So one of the most remarkable species is the bar-headed goose. So the zoological name for that is Anser indicus, which in ways but physiologists still do not fully comprehend, is able to fly without an oxygen mask over the crests of the Himalayas at 9,000 meters. We have certain water-breathing animals. Typically, they face a substantially greater challenge to obtain oxygen than air-breathing animals do, because the supply of oxygen for water breathers is the oxygen that's dissolved in water. And the solubility of oxygen in water is not high. Because of the low solubility of oxygen, water contains much less oxygen per liter. So, but in short, here we have to say that this oxygen demand for the animals is also, uh, animals are dependent on that. So different animals have different adaptations for this oxygen poor environment. Here you can see the density layering that can cut off the oxygen supply to the deep waters of a lake. So this graph here shows the oxygen that enters the lake only near the surface through photosynthesis or aeration. So x-axis here is the oxygen concentration. You can see. So when a sun-heated surface layer forms in the summer, it tends to float on top of the deep and cold layers. Little mixing between the two layers occurs through thermocline in the transition layer where the temperature changes rapidly with depth. Also, the failure of the surface and deep layers to mix cuts of the oxygen supply to the deep waters. So this red line here represents the oxygen concentration. That means the warm, low density layer of water, oxygen rich. For animals in deep waters to survive, they must be able to tolerate low oxygen levels, or they must temporarily move to other places where there is more oxygen available. In this slide, we will talk about the sea star and the corals that are in this ocean scene. They have body fluids that are similar to seawater in their total concentration of salts, although the body fluids of the fish are more dilute than the seawater. So actually this is about the sea star, corals, and the water. So here the environment is water and the animals are like sea stars. We have the coral reefs. So water is the universal solvent. We all know that. Therefore, it's required for blood and all other body fluids to have their proper compositions. So this water is actually important for animal because water is bound to proteins and other macromolecules, as water of hydration is often required for the macromolecules to maintain their proper chemical and functional properties. So animal life began in Earth's greatest watery environment, the oceans. So that's their ancestors never left the seas from the time that animal life began. And thus the salinity of the oceans has been a perennial feature for their environment. So the blood of these invertebrates actually, like you can see the coral reefs and these sea stars, they, although differing a bit from seawater in composition, is similar to seawater in its total salt concentration. So these animals therefore do not tend to gain much water from their environment by osmosis, nor they tend to lose water from their body to the seawater. So because of this situation, it's almost universal among these animals. We believe it's the primordial condition of animal life. Thus, we believe that for much of its evolutionary history, animal life lived in a setting where, for example, water was the animal. You will see the animal life mostly where the water is abundant in the environment, and there are little danger exists for an animal to be either dehydrated or overhydrated. So uh, those are the two reasons. So they believe that much of its evolutionary history of animal life is these two reasons. So that was all about the chapter first. If you have any questions, you can just email me the questions, and we will start with our next chapter. Chapter 2, that's the molecules and cells in animal physiology. Switch. So in this chapter, we will, the name of the chapter is Molecules and Cells in the Animal Physiology. We will be talking about the different cell membranes, epithelia, enzymes, and all other things that we have here, the lipids of membranes that are structured, the diverse fluid and responsive that are to some environmental factors, proteins that enter membranes with the numerous functional capacities, carbohydrates that play an important role in membranes. We will talk about the epithelia, the elements of metabolism, enzyme fundamentals, how the enzymes they catalyze the reactions in both directions. We'll talk about the evolution of enzymes, since the enzymes are the instruments of change in all time frames, the life and death of proteins, light and color, and reception and use of signals by these cells. So we will talk about that one by one. So here you can see the picture of two slow-moving predators that use their molecular weapons to capture fast-moving prey. So a cone snail sits virtually motionless in the coral reef ecosystem. You can see here. So it occupies. So yet feeds the routinely on fish. The puff adder is a notoriously sluggish but deadly African snake that resembles rattlesnakes in the strategy of hunting small mammals. So both these predators, the snail that lives in the coral reefs and this sluggish snake that resembles the rattlesnake. So this rattlesnake actually hunts the small mammals. So both predators produce the venoms that disrupt vital molecular structures or mechanisms. So the two slow-moving animals pictured here are able to consume the fast-moving prey because they have evolved ways to defeat the function of essential molecules and cellular structures in their prey. For example, we have the puff adder, that's one of the slowest moving of snakes. It feeds an animal, on fast moving rats. However, because it needs only a split second of contact with its prey to set in motion the processes that will destroy key molecular cellular properties on which a rat depends for life. So like rattlesnakes and other adders, the puff adder sits and waits for an unsuspecting animal to come close enough for a strike. So it then lunges off its victim, sinks its fangs in, and in less than 1 to 2 seconds, it injects a complex mix of compounds that attack the critical molecules in cells. So the snake then immediately releases the rat and tracks it as the rat's molecular cellular mechanism fall apart. So some of the injected compounds that strip the outer membrane from the rat's muscle cells make tiny holes in the rat's blood capillaries, permitting widespread internal hemorrhaging. So when finally the molecular cellular damage is so great that the victim can no longer function, it cannot move, the slow-moving adder moves in to eat. The second example of this slow-moving animal that consumes fast-moving prey is the cone snail that lives in the coral reef. The snail lures fish to vicinity by waving a worm-like body part that deceives the fish into coming close to eat. So the snail then harpoons the fish with a hollow barb. So these are the two mechanisms that they adapt, the snake and the snail, in order to eat their victim. So they actually make their bodies like snail. It makes some body movements, that the fake movements that can help him in eating that fish. And fish comes closer and the snail can eat that. So coming to the structure of a cell membrane primarily consists of two layers of phospholipid molecules with protein molecules embedded and attached intracellular membranes. They also have a structure-based protein embedded in a phospholipid bilayer. So the main goal actually of this chapter is to discuss the fundamental structural and functional properties of molecules and cells. So many of the properties discussed here will come up in more specific ways through the book. So 4 topics that we will be mostly giving attention will be the cell membranes and intracellular membranes, the epithelia, enzymes, how they function, their diversity and evolution, and also the mechanism by which the cells receive and act on signals. So starting with the structure of a cell membrane, so each animal cell is enclosed in a cell membrane. We call it as a plasma membrane. So each cell includes many sorts of intracellular membranes. That's the subcellular membranes such as the endoplasmic reticulum, the inner and outer membranes of each mitochondrion, and 2 closely associated membranes that form the nuclear envelope. So these membranes are thin, measuring about 6 to 8 nanometers. So cell membrane is ordinarily composed primarily of a bilayer, double layer of phospholipid molecules, in which the protein molecules are actually embedded. So similarly, the fundamental structure of intracellular membranes is also a bilayer of phospholipid molecule with protein molecules embedded in it. So principal weapon in the complex venom, like we talked about the puff adder or rattlesnake, is a set of enzymes known as the phospholipases, which break up the phospholipids. So among other effects, these enzymes destroy the phospholipid matrix in the cell membranes of a victim. That was a rat. Skeletal muscles, and thereby exposing the inside of the cells, setting membrane proteins adrift and wreaking other havocs. So phospholipid molecules are often described as having a polar head and 2 non-polar tails joined by way of ester linkages to the glycerol. So here you can see that they are, like in picture A, you can see the full chemical structure of a particular phospholipid, a phosphatidyl that's common in animal cell membranes because many different chemical structures can occupy the 2 tail positions and the labeled position in the head. Hundreds of kinds of membranes, the phospholipid molecules are possible. Any particular membrane typically consists mainly different kinds of phospholipid molecules, and the kinds may change from time to time. In the picture B, which is a model of phospholipid molecule, it shows the way that a phospholipid molecule is usually symbolized to emphasize its polar head and lipid tail. So it has a polar head that's hydrophilic and non-polar tail that's hydrophobic. So I'm talking about this model of a phospholipid molecule, the head and the tail. The head is hydrophilic head and the tail is non-polar hydrophobic tail. In figure C here, you can see that phospholipid molecules, they assemble into a bilayer with water on either sides. So when placed in water, the phospholipid molecules are spontaneously assembled into a bilayer in which the non-polar hydrophobic tail occupy the core and the polar hydrophobic head, hydrophilic head, it occupies the surface. So talking about the simple epithelia, we have here the generalized epithelium in this figure, where the cell membrane of each epithelial cells has an apical region that faces out from the underlying tissue into the cavity or open space, you can see. And there is this basal region that faces towards the tissue to which the epithelium is attached. So these are here the intracellular spaces. This is the nucleus. These are the blood capillaries. This is the basement membrane here. So this sheet actually, which is called the basement membrane, is composed of glycoproteins and particular type of collagen. So the basement membrane, I'm talking here about this. So that's composed of the glycoproteins and particular types of collagen. It's secreted mostly by the epithelial cells, although the underlying cells, they also contribute. So some epithelia are classified. So we have different kinds of epithelial cells. Here you can see the shape of the cells. So some are pyramidal, some are squamous, or some are columnar, some are cuboidal. So these are the different kind of epithelial cells. So it's this basement membrane, which is made mostly composed of these glycoproteins, they are secreted actually by those epithelial cells. The cells in a squamous epithelium, they are low and flat, whereas those in a columnar epithelium, they are high relative to their basal dimensions, like you can see here in the figure. So instead, epithelial cells, they exchange oxygen, carbon dioxide, and other materials through the underlying basement membrane with blood capillaries. You can see here the blood capillaries that are located on the opposite side of the basement membrane. So columnar epithelium actually that lines the small intestine, that's the midgut of a mammal. You can see here columnar epithelium, it mostly lines the midgut of a mammal, that's the small intestine, is a type of simple epithelium that will be featured prominently in this book in our chapter 5 and 6, and that introduces the additional aspects of epithelial morphology and function. So apical surfaces of the cells in this epithelium, they face into the lumen, open central cavity of the intestines. As digestion occurs, the liberating food molecules from foods, the molecules pass through the epithelium and basement membrane to reach the blood vessels and lymph passages that transport them to the rest of the body. So this intestinal epithelium illustrates that a simple epithelium can consist of 2 or more cell types, whereas the epithelium, which is mostly composed of absorptive digestive cells, it also includes the endocrine cells and additional cell types. Like here you can see. So intestinal epithelium also illustrates the microvilli. You all know that these projections here, they are the microvilli. These are the common feature of epithelial cells. So they are exceedingly fine finger-like projections, the microvillus of the apical cell membranes. So in the intestinal epithelium, these microvilli, they greatly increase the area of contact actually between the epithelial cells and the contents of the gut. So microvilli are often described collectively as a brush border. So collectively we call them as a brush border, all these microvilli, because they look like the bristles on a brush when they are viewed microscopically. Another significant aspect of diversity in simple epithelia arises from the geometric arrangement of cells. For example, we have the tubules or follicles that are formed by the simple epithelia. So you can see here that they are often formed by the wrapping of a simple epithelium into a closed curve and supported by the basement membrane on the outside. So a tubule formed by the cuboidal epithelium bearing microvilli forms the proximal region of the mammalian nephron. Those are the nephrons that are the kidney tubules. For example, you can see here in this picture B. So these are the cuboidal cells of the epithelium that display 2 signature properties of high metabolic activity on the apical surfaces and abundant mitochondria. So vertebrates, blood capillaries are an essentially important. For example, each blood capillary consists of single layer of highly flattened epithelial cells. So this is the mammalian blood capillary cross section here. Though three cells can be seen, all three have nuclei, but because the cells are so highly flattened that they extend considerable distances along the length of capillary. So the nuclei of two of these cells are actually positioned outside the plane of this section. So these are composed of the flattened squamous cells of epithelium. They lack the microvilli. So here in this slide, there are the types of junctions between the cells. So at a pore in a gap junction, each cell has a ring of 6 proteins that together form the pores. And the rings of the two cells, they line up to create continuity between the cells. So each ring of these proteins is called a connexon. So this is actually the connection that's made between the two cells. So tight junctions and septate junctions, they occlude the intracellular space between two cells, because not only do the cell membranes meet or fuse at these junctions, but also continuous bands around the cells. In tight junctions, the cell membranes of the two cells make contact at the ridges. So like here you can see that in this image, so these are actually called the connecting proteins. That means that these all form the junctions that we have discussed within a gap junction. So there are open pores between these cells. So at these pores, which are formed by these proteins, the connecting proteins, the two adjacent cells lack the cell membrane boundaries, and there is connectivity. Or we can say there's some continuity between the cytoplasm of the cells. So the two regions of the cell membrane therefore have different sets of channels. That's the transport membranes, the enzymes, and other classes of membrane proteins. And they are functionally different in many ways. So these differences exist between the apical and basolateral regions in the membrane, phospholipids composing the outer but not the inner leaflet of the cell membrane. So one of the important functions of epithelium is to control and mediate the transport of these substances between the apical and the basal sites of the epithelium. And thus, between different body, these regions, these substances such as anions or ions, the nutrient molecules or water, they pass through a simple epithelium by two types of paths. One is called the transcellular path and other is the paracellular paths. So these are actually the two paths from which these substances, for example, ions or nutrient molecules or water, they pass. So transcellular is one term for that, and another is the paracellular. So what actually happens with the tight junctions, they interfere with block or the paracellular movement of substances across an epithelium. So in some epithelia, the tight junctions, they almost prevent all paracellular movement. So in others, however, the tight junctions permit the extensive paracellular. So their functions are actually different in sorts of molecules or ions, and the epithelia are described as the leaky. So when they permit this extensive paracellular movement, these molecules or ions, the epithelia that control this kind of movement is called as leaky. So a substance that crosses an epithelia by a transcellular path must pass through two cell membranes. So talking about the enzyme fundamentals here in a story of like the celebrated jumping frog of Calaveras County, Mark Twain was a scientist. He appealed to the imagination of his readers by extolling the awesome jumping abilities of a frog, probably a common leopard frog, like you can see here in the picture. So leopard frog, whose zoological name is Rana papiens, named Dan Webster. Anyone who has ever tried to catch leopard frogs knows that when first disturbed, they hop away at lightning speed. So thus it's very hard to smile in and knowing admirations, as Twain describes Dan Webster celebrating jumping feats. So muscles actually can work only as fast as they are supplied with adenosine triphosphate. So amphibians, however, have only modest abilities to make ATP, that's the adenosine triphosphate, using oxygen, because they have relatively simple lungs and can supply their cells with oxygen only slowly. So for leopard frogs to hop along as fast as they do when fleeing danger, they need to make ATP faster than the oxygen supply to their muscles permits. So this is the physiological function. That means the production of this ATP, the adenosine triphosphate, that gives them energy to move faster than the oxygen supply to their muscles permits. So for leopard frogs to hop along as fast as they do fleeing, so they need this ATP, the adenosine triphosphate. So that's why they must take ATP by anaerobic mechanisms, not requiring oxygen. A crucial reason they can do this is that their leg muscles are well endowed with the enzyme. So they have a particular enzyme for that. That's the lactate dehydrogenase. So that's an enzyme that actually helps him in production of this ATP. So compared with the leopard frog toads, such as the common western toad of North America, we have here this, which is also called the uh, Bifoborios. They are not nearly as well endowed with lactate dehydrogenase enzyme. So thus they cannot make ATP to a great extent without oxygen. And the slow rate of oxygen delivery to their muscles means a slow rate of ATP production. So this means that this rate of oxygen delivery to their muscles and ATP production, they are actually interlinked. So the absence of this lactase dehydrogenase enzyme present in this kind of west toad of North America, that means as there will be a slow rate of ATP production, explaining that why they cannot hop as long as fast as frogs. So what actually enzymes are? They are actually the protein catalysts that play important roles. So they actually speed the chemical reactions and often regulate the reactions. So these are the two important functions that are formed by these enzymes. So enzymes are protein catalysts that play two important roles. That means they speed up the chemical reaction that's going on, and they also often regulate that reaction. So when we say that an enzyme, so we might say that this enzyme is actually a catalyst. Catalyst means that any compound that can increase the rate of reaction or accelerate the rate of reaction without undergoing any change in itself. So the enzyme remains as it is, but it increases the rate of reaction. So we mean that it's a molecule that accelerates the reaction without changing, without any changes at the end in itself, being altered itself. So this reaction catalyzed by the lactic dehydrogenase (LDH) in case of frogs is important for escape by the frogs. So it's the reduction of pyruvic acid to form lactic acid, a reaction in which each pyruvic acid molecule is combined with these two hydrogen, in which these pyruvic acids are combined with two hydrogen atoms. So although the presence of this LDH, that means the lactic dehydrogenase, it speeds their reaction, it's not itself altered. So this is an enzyme. So enzymes, they speed up the reaction, like here in lactic dehydrogenase, it speeds up this pyruvic acid, is produced from glucose by glycolysis, and then it's broken down to lactic acid, and then this pyruvic acid here, you can see it's broken down to lactic acid by means of this lactate dehydrogenase enzyme by the use of two hydrogen atoms. So when this reversible reaction proceeds from left to right, pyruvic acid is reduced by receiving two hydrogen atoms from NADH2, yielding lactic acid and nicotinamide adenine. So that means the lactic dehydrogenase that was used, the enzyme that was used in this chemical reaction, it actually catalyzed the reaction without undergoing change in itself. So it's actually considered as an enzyme. So enzymes actually accelerate the reactions by lowering the needed activation energy. So in this graph here, like you can see, so catalytic effectiveness of an enzyme depends partly on the activation energy of the enzyme catalyzes the reaction. To understand the implications of this activation energy, it's necessary to recognize that a substrate molecule must pass through an intermediate chemical state that's termed as a transition state. So this activation energy is very necessary for increasing the energy that's required to achieve the transition state without catalysis. So moving to the bioluminescence and fluorescence in Aquaria victoria, jellyfish found along the west coast of United States. We will talk about this. So we cannot talk in detail because we have less time. So we have to just finish the other chapter also. So we will just try to finish this. I will try to give you some idea about how this bioluminescence and fluorescence in the jellyfish takes place. Hello? So the ability of cells to produce the light biochemically, that's called the bioluminescence, is widespread in animals. So it's most common in the ocean where the bioluminescent species are found in most of the principal animal groups. So including many invertebrate groups and fish, as well as bacteria and protists. For example, the bioluminescence must be distinguished from the fluorescence. So these are the two terms actually. So both processes can occur within a photocyte. So in bioluminescence, light is produced de novo. In fluorescence, light is not produced de novo. Instead, pre-existing light is absorbed and re-emitted at a longer wavelength. So we have the green fluorescent protein, that's GFP, that discovered in a bioluminescent species of jellyfish, genus Aquaria. So this provides a prominent example of fluorescence. So how it happens is that the photocytes in the jellyfish have a biochemical pathway that in isolation generates the light of blue wavelengths. So in the intact cells, however, the pathway is intimately associated with this GFP, that's the green fluorescent protein. And the final step of light production is the energy from the light producing pathway that's produced by the resonance. So a radiation less processed to the GFP emits light at green wavelengths. So the cluster of photocytes on the margin of the bell of the jellyfish, here you can see. So on the margin of this jellyfish, so there is a cluster of photocytes that makes this jellyfish to glow green. So talking about the squid, the ecological function of the symbiosis for the squid, we have the bobtail squids, that's the Eperamina scolopus, that forms a lifelong symbiotic relationship with the gram-negative bioluminescent bacterium. So here the bacteria in the light organ in the center of the mantle cavity of the squid emit bioluminescent light downward so that the night the squid blends in with moon or straight when viewed from below. Soon after a squid hatches, the Vibrio fisheries bacteria specially attached to the key epithelial surface that the bacteria after they are acquired promptly induced to be lost simultaneously. So here you can see the picture, it shows the bacteria that enter the ciliated pores to populate the squid's light organ. So careful studies have revealed that this recruitment, the formation of this symbiosis, entails an intimate relation between the squid and the bacteria. So a young squid represents specialized epithelia to its seawater environment to acquire the specific bacterial symbionts, which populate deep crypts within the squid's light organ. So once this acquired, the symbionts initiate the lifelong loss of those epithelia, making further acquisition. So that was all about the acquisition of these bacterial symbionts. So we also have the pigment aggregation and dispersal within the black pigmented chromatophores from the skin of a codfish here. So that was all about the chapter second, and I will just give you a brief idea about the chapter third. So we will be talking about the osmosis, diffusion, how the transport of the solutes. So this is actually the chapter third for this lecture. So we will be talking about the osmosis and how the diffusion of and how the transport of solutes and water takes place. So passive solute transport by simple diffusion and concentration gradients that give rise to most elementary form of simple solute for diffusion. We will talk about the electrical gradients, the biological aspects of diffusion, passive solute transport, and osmotic pressure. Some osmotic pressures can be measured in several ways. We'll talk about the osmosis, hydrostatic pressures, and aquaporins, and osmosis and solute physiology that are in contact. So for example, the mechanism that must exist to transport sugars from the open central cavity of a hummingbird intestines into its blood following a meal. How this happens is that hummingbirds are dietary specialists. They feed almost exclusively on flower nectars, which are principally solution of sucrose and other sugars. So notably the glucose and fructose. So when sucrose itself is digested, it yields the glucose and fructose. So thus a hummingbird has an abundance of glucose and fructose in its intestines. So after digesting a meal, and these simple sugars represent its immediate reward for feeding. So these sugars actually are in solution in the lumen, so hollow central core of the intestines, and for them to do the bird any good. So they must move into the bloodstream to be carried throughout the body. So how this happens, we have first imagined that glucose and fructose in the lumen of hummingbirds' intestines could simply diffuse across the intestinal epithelium into the blood vessels. So glucose and fructose are hydrophilic, and because hydrophilic molecules tend to mingle with the hydrophobic ones, the sugars can be expected to have difficulty dissolving in the highly hydrophobic interior of the apical and basolateral cell membranes of the epithelial cells. So sometimes the glucose and fructose from a meal cannot diffuse into hummingbirds' bloodstream significant rates. How do these sugars move into the blood? So questions of this sort are among the most common and important that are faced in this study of animal physiology. So there are three focal examples in which the transport occurs. One is the single animal cell that's related to ion concentration inside and outside. You can see here. The extracellular fluid, we have sodium, potassium, chloride, and intracellular, we have these. So this is the size of the lettering symbolizes here the relative ion concentration inside and outside the cell. So sodium, potassium, and chloride differ substantially in their concentrations inside and outside. So these are the anionic proteins here. So in the three examples in which the transport is important to animals, we'll have defined the subject. So for example, one of our focal examples is a single cell inside an animal's body. So a typical animal cell is passed by the extracellular fluid, We can think of the cell membrane as separating an intracellular fluid, the inside from the extracellular fluid on the outside. The solutions on the inside and outside of a cell are similar in their sum total concentrations. So our second focal example is the outer gill membrane of a freshwater fish. Here you can see that's living in a pond or a stream. So the fluid portion of the blood of a freshwater fish's blood plasma is more concentrated in sodium and chloride ions and the inner other inorganic ions than the surrounding pond or stream. So our third example is the epithelium of the small intestine of a bird or a mammal. Here you can see the epithelium consists of single layer of cells bearing microvilli on their this. As I stressed already in our discussion that hummingbirds, they dissolve the sugar molecules such as glucose and fructose and must cross the epithelium from the intestinal lumen. So these are the three examples, focal examples in which the transport occurs. So simple diffusion, here in this diagram, you can see that the simple diffusion that's viewed macroscopically and microscopically. For example, in picture A here, you can see a beaker that's divided in half by this porous artificial membrane that permits the passage of glucose molecules, but nothing else. Solutions on either side of the membrane differ in their glucose concentration. And in the picture B, looking at the same system microscopically reveals that random motion are more likely to carry this glucose molecular pores in, that occur in artificial membranes, but not in living biological membranes. So what actually the passive solute transport takes place, how actually the passive transport takes place by simple diffusion. It's to understand this simple diffusion, for example, consider a beaker divided by a non-living membrane that's penetrated by many microscopic pores through which the sugar glucose, but nothing else can pass. So a solution of glucose in water is placed on each side of the membrane, with the solution on the left having a higher glucose concentration than that on having a lower. So since we all know that diffusion is the moment of, here will be the moment of the glucose molecules from higher to lower concentration. So there will be a gap with the artificial membrane. So however, that's the mechanism of motion that carries these glucose from the left to right. That means from higher concentration to the lower concentration. So this mechanism of glucose transport that we have just described in simple diffusion transport that arises from the molecular agitation exists in all systems above absolute zero and from the simple statistical analysis. So this is actually very important for the diffusion of these glucose molecules from higher to lower concentration. So time required for diffusion through water to have a concentration difference. So it depends upon the thickness of a cell membrane, the radius of a small mammalian cell. For example, the thickness of human heart muscles, which is 2 centimeters, it will take 4.7 days. And it also depends upon the length of a long human nerve cell. Present there. So diffusional concentration of a solute in a boundary layer next to an animal, or we can say uh, to an animal or a cell. So there are electrical gradients that often influence the diffusion of charged solutes at the membranes. For example, many solutes of biological importance, such as sodium positive, or we can say chloride and other inorganic ions, they bear an electrical charge. So diffusion of these charged solutes, in addition to being affected by concentration gradients, is influenced by the forces of electrical attraction or repulsion forces that do not affect the movement of these uncharged solutes such as glucose. Like you can see here in the figure, that to understand why such electrostatic effects need to be considered is the analysis of diffusion, which is a key principle that solutions at large are electrically neutral. So in bulk solutions, for example, of solution that is away from contact with the membrane, the concentration of positive and negative charge are always equal. So the net charge in any region of bulk solution is therefore zero. So you can see here that within a few nanometers of a cell membrane or epithelium, the solution on one side of may have a net positive charge, because positive ions of these because the positive ions, they outnumber the negative ones. So conversely, within a few nanometers of the membrane epithelium, solution on the other side may have a negative charge, because the negative ions, they outnumber the positive ones. So under these circumstances, we can see the diffusion of electrically charged solutes may be affected by the electrical attraction, or we can say the repulsion. So electroneutral active transport is also responsible for secretion of stomach acid in the vertebrate stomach lining. We will talk about this, that how the proton pump secretes stomach acid, which is a protein that's expressed in parietal cells. So the pump actually exchanged these two proton ions for two potassium ions during each pumping cycle. So it's thus electroneutral. So apical membrane at each parietal cell projects into the cell as invaginations called the canaliculi, or the proton pump also helps in protein molecules are positioned in the portions of the apical membrane that line this canaliculi. So this is actually the responsible for secretion of this stomach acid in the vertebrate stomach. So summary of the active and passive ion transport in a typical animal cell. So active transport, the animals are able to transport many solutes across their cell membranes and epithelia directions away from electrochemical equilibrium. So these mechanisms are capable of such transport are called as active transport mechanism, or we can say these are actually the pumps. So like we already discussed about the proton pump, this active transport mechanism transports the ions or that are able to create a voltage difference across a membrane. So because of this mechanism, move charges, active ion transport mechanisms do not necessarily generate the voltage differences. However, because they often simultaneously transport two ions, and the transport pump, informally known as the proton pump. So primary and secondary active transfer differ in their cellular molecular mechanisms. So we conclude that the cell must transport the potassium positive outward and K positive inward, carries out this transport. So this is again is the electrochemical. So if it is to maintain observed differences of ion composition across the cell membrane between the intracellular and extracellular fluids, the sodium and potassium ions carry out this transport using ATP bond energy. So this actually summarizes the processes of both active and passive ion transport between the animal cell and the extracellular fluids that bath in it. So two examples of the energy coupling via an intermediate step in which the energy is temporarily stored as potential energy. So There was a scientist, that's Peter Mitchell. He revolutionized the knowledge of energy metabolism in cells by proposing an entirely novel idea for a mechanism by which the cells can couple these energy sources to work performing processes. So he was the Nobel Prize winner for this idea, termed the chemiosmotic hypothesis in certain contexts. So to see the underlying concept, for example, in this figure, you can see, consider a type of mechanism that engineers sometimes employ for energy coupling. At the left in part first of the figure, here is the part first of the figure, there is a power plant. At the right is an apparatus that does work by use of energy. So question to be addressed is how energy from the power plant can be transported, can be coupled to work performing apparatus. So approach shown in first is to store energy from the power plant in the form of potential energy, then allow the work performing apparatus to draw this energy from the potential energy. So living organisms started to employ similar energy coupling systems almost from the start of their evolution. So these energy coupling systems do not use the elevation of water to store this potential energy instead. So these here, the power plant is actually the bonds of ATP or food molecules. And then the output or the work done is the proteins that couples ion diffusion to performance of work. So this is the similar examples to the living membranes. So this is the magnified view of two solutions that are similar in colligative properties. So how we can measure the osmotic pressure of a solution directly? We have a piston osmometer, and then in the figure B, the consequence of if the piston is free to move. Here, the consequence of the solution is placed under the hydrostatic pressure that exactly opposes this osmosis. So this is actually the solution to be measured, which is separated from pure water by a semi-permeable membrane. So we all know that semi-permeable membrane allows only water to pass through it. That forms the end of an ideal frictional piston. So how to measure this osmotic pressure of a solution directly is for, we all know that physical chemists, they have devised methods of measuring this rather than calculating it from other colligative properties. So these methods are the gold standard of osmotic pressure measurements, but usually they are not employed for day-to-day practical work by biologists because they are very cumbersome to carry out. So figure here, it illustrates that how we can measure the osmotic pressure of a solution. So the piston is placed in water. The end of the piston consists of semi-permeable membrane. The reason for using such membrane is that it ensures that the solution will not be corrupted by losing some of the solute during the measurement process. So this piston movement can be prevented by exerting a force on the piston as shown in figure. The force will raise the hydrostatic pressure in the solution. So it's higher than that in the pure water. So the elevated hydrostatic pressure in the solution will then tend to force this water molecules through pores of the membrane by pressure-driven streaming bulk flow called ultrafiltration. So knowing this, you could then accurately predict that if the two solutions A and B that are placed on the opposite sides of a water permeable membrane, the direction of water transport by osmosis will be from A to B. Recall that we know that here you can see that direction will be from A to B, as we have seen that it's the osmotic pressure that controls this. So recall that one way to define osmotic pressure is that is the property of a solution that allows one to predict whether the solution will gain or lose the water by osmosis when it undergoes osmotic changes with other solutions. So the physical chemist's approach permits measurement of this property and is in fact the most fundamental ways to ensure the osmotic pressure. So the measurement of osmotic pressure, biologists obtain using freezing point osmometers or vapor pressure osmometers. They typically provide the same information by different means. So this is the figure in this here shows the osmotic uptake of water by a freshwater animal, because the freshwater animals have the osmotic pressures in their blood that exceed the osmotic pressure of fresh water. So water molecules moving through a single aquapore one at a time. For example, here is an aquapore monomer with a single membrane that's modeled by means of computational biophysics. So blue represents the water molecules, the layers of water molecules at the top, and bottom represent the fluids outside and inside a cell. So water molecules actually passing through the aquaporin pore are represented at a larger size than the other water molecules. So that was all about today's lectures, today's chapters. So if you have any questions, you can just send me an email so that I will just try to um, answer your emails ASAP. Any questions about the syllabus, about the assignments, you can just forward me all through emails. Thank you so much.